Now, the topic on chapter 7 is on microbial physiology and genetics. So, the learning objectives for this chapter, we have to define what are the prototropes, chemotropes, autotropes, heterotropes, photoautotrope, chemoheterotropes, endozyme, exozyme, plasmid, or the R factor, which are found in chapter 7. Differentiate between catabolism and anabolism. Explain the role of adenosine triphosphate molecules in metabolism, and briefly describe each of the following biochemical pathways such as aerobic respiration, glycolysis, the Krebs cycle, or we call it the TCA, tricarboxylic acid cycle, and the electron transport chain happens in bacteria, and oxidation reductions and photosynthesis. This is more on the plants. Now, in microbial physiology, it is important that you know the physiological action of some bacteria. That some bacteria need some nutritional factors, or the survival of the bacteria would also depend on the environmental factors for their growth. Then, in the laboratory, we are controlling the microbial growth by putting them in a certain pure cultures. Now, this pure culture discussion will be transferred next week or a part of your laboratory topic. Kasi maraming klaseng culture media in a certain type of bacteria that will be used on a certain type of experiments. Then microbial metabolism. Now let's talk on the growth of microorganisms. Now the growth of the microorganisms, like the eukaryotes, follow certain nutritional requirements in order to grow. So what are these requirements? Some requirements like the food. So they need food. What are their food? Some elements like carbon, hydrogen, oxygen, or which is the major component of the organic molecules, such as sugar, nucleic acids, amino acids, and lipids, are may considered as one of the food needed in the microbial growth or the bacteria. Nitrogen source is a good source of amino acids. So amino acids and proteins contain some elements of nitrogen. Sulfur, since sulfur was embedded in some amino acids. Even in phosphorus, phosphorus is found in nucleic acid and some in ATP. Or some may be found also in the membrane lipids. While the potassium, magnesium, calcium, and iron could be found in my enzyme and cofactors that will be performing or hastens the reaction during metabolic pathway. Now, growth factors, so compounds that bacteria requires but cannot synthesize. Growth factors also include the environmental conditions, some chemical conditions in the environment, and the temperature, the amount of energy, such as sunlight or some amount of oxygen in the area. But there are some bacteria that don't like oxygen, and they called it anaerobic bacteria. So kung aerobic bacteria, these are bacteria that prefer in an environment that has oxygen, and they use this oxygen in their metabolic pathway. Now, different or various type of bacteria have different requirement. So let's have the photoautotropes. They utilize their synthesis or the raw material. They capture the light as the source in their synthesis, like in the plants, they convert light into organic molecule. So in the process of photosynthesis, there are some bacteria that carries pigments like cyanobacteria. Cyanobacteria has the ability to perform photosynthesis. Photoheterotropes, when we say photoheterotropes, they are capable in photosynthesizing. At the same time, they acquire nutrients or energy from other organisms. Chemoautotrophs, they utilize inorganic or organic molecules. Hindi necessary the light kasi kung light photo, kung chemo, they utilize carbon dioxide, nitrogen, nitrates. Like for example, the denitrifying bacteria, nitrifying bacteria. So they utilize that one and synthesize to produce another molecule. So those are chemoautotrophs. When we say chemoheterotrophs, they acquire the elements at the same time they acquire another energy from other organisms. Okay, so for example of organisms, including also some eukaryotes that perform this kind of food acquiring behavior, we have the photoautotrophs usually performed by plants, algae, some protozoans that contains pigments. Cyanobacteria is another prokaryote that perform this kind of synthesis. Chemoautotrophs, bacteria. Sa heterotrophs naman, the good example is humans, animals, and some bacteria also. So the common sources of bacteria used in the synthesis, we have the nitrogen source. So ammonium is used to solve nitrogen source by most microorganisms. So ammonium could be produced by nitrogen gas, so through nitrogen fixation. So nitrogen gas is acquired in the atmosphere. So there are some bacteria that converts the nitrogen into ammonium or to form to nitrate or nitrite. And makikita yan sila sa mga soil. Example, ito yung mga bacteria na makikita sa soil. Sulfur source, and they are important also in the nutrient cycle because this will help the plant to take in nitrogen and transport it, convert it into protein. So without this, nitrogen or no production of protein within the plant tissue. So wala na tayo protein. Dili man ta ka-intake of protein physically because nitrogenous gas is fatal in our system. So kailangan ang nitrogen gas matransform siya into a different form. So sulfur source, most microorganisms use sulfate as a sulfur source. So some are phosphate used as phosphorus source. So the microorganisms, they are very essential in converting these elements to be used by another macroorganisms. So sa food chain, especially sa nutrient cycle, they are essential because they're the first group of organisms that will convert the elements or mga inorganic compounds into a usable, consumable compounds needed by the macro animals or the macroorganisms. Now, in terms of environmental factors for microbial growth, magvavary din ang different species of bacteria. Not all bacteria favors the optimum range of temperature or the pH or a very saline solution na aquatic environment. Now, different species have their own environmental requirements, such as in terms of temperature, there are species may vary to different temperature requirement. We have psychrophile. Psychrophile thrive between the temperature 13 degrees Celsius to 20 degrees Celsius. So medyo malamig ito. Mesophile, these are bacteria that thrive between 30 degrees Celsius to 37 degrees Celsius. So muna ang optimum temperature range. Excuse me. And thermophile, these are bacteria that thrive in a very hot condition. So 50 to 60 degrees Celsius. But there are still some bacteria, even in the volcanic area, may magta-thrive na bacteria rin. And yun yung mga examples of thermophiles. Now, in terms of pH, some thrive in neutral environments, ranges from 6 to 8. Acidophile, pH is 1 to 5. Then alkalifile, or yung pinaka-basic na environment, they thrive mostly sa 9 to 11 na pH. And in terms of oxygen availability, meron ding mga bacteria like obligate aerobe, thrive in availability of oxygen, obligate anaerobe and facultative anaerobe usually don't thrive in, or not, usually they could be found sa mga vacuum space where oxygen is absent. Then we have microaerophile, where 5 to 10% of oxygen in the environment. And in terms of water availability, or the type of, not water availability, but the osmolarity of the water, kasi a concentration of solute in the water, we have the osmophile. So when we say osmophile, they are adapted to the environment which are high osmotic pressure. So daghang solute sa aquatic solution, like a higher concentration of sugar. And in terms of halophile, these are bacteria that thrive in saline environment. So medyo salty environment. Now, bacteria could be cultured in artificial environment through culture media. So culture media may come in different forms like nutrient agar, or solid, or in liquid. So broth in liquid form, nutrient agar in a solid form, and some may also do the semi-solid. Now, in terms of the content in a media, may vary also. One good example, we have blood agar or chocolate that is more for a certain type of group of bacteria. Later on in chapter 8, this will be explained the usage of different types of culture media. So we have the common one, yung nutrient, agar, and the broth. And we have the selective media in which you're going to test the bacterial growth using an inhibitor. And we have differential media, substances that certain bacteria change, recognizable by way we added a certain type of, i-observe mo ang growth of bacteria when you added a certain chemical towards the culture. So nutrient broth only contains peptone, meat extract, and water. And this could be substituted also at home. Sa glucose naman, ito yung content ng glucose sa nutrient agar. So in a common culture media, since this one, ang culture glucose, wala masyadong protein, we add up the nutrient broth para maging source of food ng bacteria. Now, bacterial growth in the laboratory conditions may follow certain patterns. That's why in observing a colony of bacteria may takes only 24 hours. So dapat 24, some 12 hours. Munang in the laboratories, microbiologists will stay 24 hours to observe the culture growth. Kasi kung mugubiyan pa siya sa 24 hours, dili na siya viable, dili na niya murag kumbaga nanay mahitabo ana. Because in this growth phase, first we have the lag phase, then lag exponential phase and stationary phase, then the downside of it we have the death phase. So ito yung crucial sa mga microbiologists in observing culture media growth. That's why they observe it from 12 to 24 hours. Beyond that, dili na. You have to redo na naman the experiment to observe the growth. So bacteria multiply in binary fission since they don't have sexual or gametes to reproduce. The cell itself divides through binary fission. So in terms of the phases of growth in bacteria in the culture, the lag phase is the first phase where the bacteria synthesize the macromolecules required for multiplication. So this is the time where the bacteria use the nutrients in the culture and slowly divides itself through binary fission. Then the log exponential phase, this is the doubling time measured during this period. So the bacteria is actively dividing itself to different several cells through binary fission, paspas ang iyang ano. And the stationary, this is the space where the bacteria have already occupied the culture and almost utilized all the nutrients in the culture. Then the death phase, this is the time the nutrients in the culture was already been utilized, so the bacteria slowly to die out. So some cells are already dead kasi wala na silang pagkain. Almost na kain na nila yung mga nutrients in the culture. Now, let's proceed sa bacterial metabolism. So sa bacterial metabolism, we have here the term anabolism and catabolism. So when we say anabolism, assimilation, energy requiring, and catabolism is dismulation or energy acquiring, sa anabolism, energy requiring. So in this process, it involves yung different metabolic pathways. We have the glycolysis, the pentose phosphate pathway, the Krebs cycle or the TCA cycle, respiration or aerobic or anaerobic respiration, and one example is the fermentation. Ang aerobic respiration is the glycolysis, and anaerobic respiration is represented by fermentation. And we have also the electron transport chain happens in the bacterial cell membrane. Since they don't have, sa eukaryotes, ETC, electron transport chain is performed in the mitochondrial double membrane, while sa bacteria, they don't have mitochondria. So, nangyayari ito sa plasma membrane mismo ng bacteria. To differentiate between the catabolism and anabolism. So, when we say catabolism, from the molecules, it breaks down into smaller particulates. So, for example, we have glucose breaks down into 3-molecule carbon chain called a pyruvate to produce ATP. While sa anabolism from smaller units, it forms into a larger molecule. So when we say kata, you have to cut and put into small pieces. When anabolism, you are going to put all the bits and bits of molecules into a larger molecule. One good example of anabolism, yung example ng anabolism, protein synthesis. Okay, protein synthesis. Dito sa table, the difference between comparison of catabolism and anabolism. So we have here purpose, energy generation and formation, useful compounds. So in terms of energetics, catabolism yields energy, while anabolism requires or uses the energy. So in the metabolism pathway, there are also combination of catabolism and anabolism because example yung Glycolysis, it uses energy to break down the carbon molecule. So it uses ATP to break down the 6 carbon atom into pyruvate. So that is example of anabolism. Then sa catabolism naman, sa glycolysis, meron din catabolism. Katong nag-produce siya og ATP. Later on, makita natin ang process or the cycle of glycolysis, TCA, and ano pa yung isa ka, Krebs cycle or TCA. So these are the metabolic pathways. So the metabolic pathways, we have the pentose pathway, where you're going to oxidize glucose to pyruvate. Actually, pentose sugar ang ginagamit instead of glucose. When we say pentose, it is a 5-carbon chain sugar. And glycolysis uses the 6-carbon atom carbohydrate. One good example is yung glucose. So we have here the pentose pathway, the glycolysis. This is the TCA below. And fermentation. So fermentation happens when no oxygen is involved. This is the example of anaerobic respiration. So the absence of oxygen. So let's have some good glucose. So glucose. So i-review natin si anabolism. Anabolism, one good example is from ATP, i-breakdown niya into ADP. So, na-simplified ang adenosine triphosphate into adenosine triphosphate. So, na-breakdown, nahiwalay sa kanya ang isang phosphate molecule. While ang catabolism, one example is from ADP to ATP, where a phosphate is added to produce. Kung tingnan natin dito, no? Hindi magkatugma ang diagram. Okay. Pero sa base sa ilang example, actually this came from 2 different resources. Ito iba sa table. Dito itong diagram, iba din ang kaniyang breaks down molecule. Tama. Okay. So during the glucose synthesis or glycolysis, okay, to break down glucose into 3-carbon pyruvate, you need ATP to break down the chain into 3-carbon atom. So, magiging glyceraldehyde nito. Then, another energy will be used by NADH. Then, the phosphate group will be removed. Then, you will be producing ATP. Then, you have the 2-pyruvate molecule. So, in 1 molecule of glucose, you're going to produce 2 molecules of pyruvate. Now, when the pyruvate undergo synthesis, it will transform into acetyl-CoA. And the acetyl-CoA is required in the TCA cycle or triglyceric acid or Krebs cycle. So TCA will enter to the acetyl-CoA will enter to the TCA cycle, transforming from citrate, going to different transformations of acetic acid. So dapat i-change ko pala ito. Nawala. Oh, yan. Producing NADH, GTP, FADH, and another NADH on the 8th step of TCA. Sige lang, dili man ako siya i-appeal sa exam, pero the production of energy in TCA is involved. So the main raw material for TCA is acetyl-CoA. Without acetyl-CoA, TCA or the Krebs cycle will not proceed. But another thing also, it requires enzymes. So the enzymes are involved in from the glucose kasi may enzyme ito pag breakdown ng bonds sa mga carbon chain. So without the enzymes, the energy will not be reproduced. So there are two molecules of carbon dioxide produced in TCA and how many ano? 1, 2, 3, 4, 5. 3 NADH, 1 FAD, and 1 GTP produced in a one molecule of acetyl-CoA. Since dalawa man ang pyruvate, times 2 lang na ninyo. So that will be the energy. So inyo na siyang i-multiply. Saan natin kung arrow? Now, let's have the electron transport chain happened in the bacteria. Uy. Ay, yeah. Asan ito?
An electron transport system consists of a series of electron carriers that sequentially transfer electrons to the carrier with the next higher reduction potential, that is, to a stronger electron acceptor. Electron flow begins with an initial electron donor, such as a carbohydrate, and ultimately transfers all electrons to the strongest electron acceptor, which in this example of aerobic respiration is molecular oxygen. During glycolysis and the tricarboxylic acid cycle, organic molecules donate electrons associated with hydrogen atoms to NAD+ molecules. The resulting NADH molecules act as electron donors to the electron transport system. A typical bacterial electron transport system used in respiration includes at least 3 functional components, beginning with a complex generally called a substrate oxidoreductase. This enzyme has a number of more specific names, including NDH1 for the complex in bacteria. NADH donates electrons to a flavin mononucleotide or FMN molecule within NDH1. As the electrons reduce FMN, the molecule also takes on 2 protons to balance the charge, becoming FMNH2. In its reduced form, FMNH2 can donate electrons one at a time to an adjacent iron-sulfur center that is a stronger electron acceptor. The iron-sulfur center aerobic respiration is molecular oxygen. During glycolysis and the tricarboxylic acid cycle, organic molecules donate electrons associated with hydrogen atoms to NAD+ molecules. The resulting NADH molecules act as electron donors to the electron transport system. A typical bacterial electron transport system used in respiration includes at least 3 functional components, beginning with a complex generally called a substrate oxidoreductase. This enzyme has a number of more specific names, including NDH1 for the complex in bacteria. NADH donates electrons to a flavin mononucleotide or FMN molecule within NDH1. As the electrons reduce FMN, the molecule also takes on 2 protons to balance the charge, becoming FMNH2. In its reduced form, FMNH2 can donate electrons one at a time to an adjacent iron-sulfur center that is a stronger electron acceptor. The iron-sulfur center passes the electron on to the next iron-sulfur center and can then receive another electron from FMN. Through a series of redox reactions, the electron passes from one center to the next in the NDH1 complex. The transfer of electrons releases energy that NDH1 can use to pump protons across the membrane. At the end of the chain, the electrons are transferred to a quinone, which picks up 2 protons from solution and is thus reduced to quinol. Quinones are generally designated Q, and quinols are designated QH2. The quinols diffuse within the membrane and carry reduction energy to other electron transport system components. This quinone pool represents the second of three functional components of the electron transport system. The third component of the electron transport chain is a terminal oxidase complex. The complex in this example is called more specifically cytochrome B-quinol oxidase. Within the complex is the binding site for quinol. Also in the complex are two iron-centered heme groups and three copper atoms. The quinol transfers its electrons to the adjacent heme group and releases two protons, usually across the membrane. Next, the electrons move from the first heme group to the second. This unique complex couples the electron transfer between the two heme groups with proton pumping. The iron and copper atoms pick up molecular oxygen and hold it in place until it receives a total of 4 electrons, only 2 of which have been generated so far. Note the energy from the 2 electrons donated by NADH fueled the pumping of 8 protons across the membrane. In order to become fully reduced, molecular oxygen requires the electrons from an additional NADH molecule. In total, the 4 electrons from 2 NADH molecules result in the pumping of 16 protons across the membrane. 4 protons from the cytoplasm join the electrons and oxygen to produce 2 molecules of water. In this way, oxygen, which has the strongest affinity for electrons of any other component of the electron transport system, is the final electron acceptor.
Okay, so that's for the electronic transport in bacterial cell. So in eukaryotes, ATC happens in the mitochondria. Since bacteria don't have mitochondria, it happens on the cell membrane. Okay. Next type of biosynthesis, we have fermentation. Fermentation is a metabolic process in which final electron transport is an organic compound. And this happens without the presence of the oxygen. So it uses carbon dioxide. So it converts pyruvate from glycolysis into lactate, so lactic acid. Some forms of products of fermentation could also produce ethanol. So from acetaldehyde or acetyl-CoA, you will have alcohol. So let's have the fermentation on alcohol.
Professor Dave here. Let's get anaerobic.
He knows a lot about the science stuff. Professor Dave explains.
In the biochemistry series, we looked at aerobic respiration. This is the process by which the cells in our body generate ATP through glycolysis, the citric acid cycle, and oxidative phosphorylation. This third step is the one that produces the bulk of the ATP payout, and it relies on the presence of molecular oxygen, because oxygen is the final electron acceptor in the electron transport chain. So without oxygen, this process cannot occur, hence the term aerobic, which means involving oxygen. But there are other methods of ATP production that do not rely on the presence of oxygen that we will want to understand now that we have begun to look at different kinds of organisms. Those would be anaerobic respiration and fermentation. The latter is used by animals such as ourselves when the body is not getting enough oxygen to fuel strenuous activity. And both processes are also used as a metabolic pathway by certain microorganisms that exist primarily in environments without oxygen. So let's get a closer look at these now. With anaerobic respiration, an electron transport chain is still used, but the final electron acceptor at the end of the chain is something other than molecular oxygen. For example, some bacteria will use a sulfate ion and produce hydrogen sulfide instead of water as a byproduct of this activity, which produces an odor of rotten eggs that you may have smelled in certain environments. Fermentation, on the other hand, does not involve respiration of any kind. This will always begin with glycolysis, just as we learned before, as this step does not require oxygen and does result in the production of 2 ATP per glucose molecule. But the pyruvate that results with oxidation by NAD+ will not feed into the citric acid cycle. Instead, something else will happen depending on which type of fermentation is occurring, so as to regenerate NAD+ from the NADH that is produced, allowing glycolysis to continue. The two types of fermentation are alcohol fermentation and lactic acid fermentation. With the first of these, glucose is oxidized over the 10 steps we learned about to produce 2 pyruvate molecules and 2 ATP molecules, using 2 molecules of NAD+ in the process. Then each pyruvate will undergo decarboxylation to produce acetaldehyde. And finally, acetaldehyde is reduced by NADH to produce ethanol. This allows for the regeneration of NAD+, which can then be used in glycolysis again. The final product, ethanol, is the kind of alcohol that humans consume, hence the term alcohol fermentation. And this process has been utilized for centuries to produce alcoholic beverages such as beer and wine. Yeast, which is a unicellular member of the fungi kingdom, performs alcohol fermentation, so this organism is used in these processes to produce the alcohol we drink, and also by bakers, where the carbon dioxide gas produced during the decarboxylation step is what causes bread to rise. Next, looking at lactic acid fermentation, again glycolysis occurs precisely as we have learned, but this time the pyruvate that is produced will not decarboxylate. Instead, it will be directly reduced by NADH to produce lactate. This again allows for the regeneration of NAD+ for use during glycolysis, and also involves no release of carbon dioxide. Lactate is the conjugate base of lactic acid, and it is the production of this compound by certain fungi and bacteria that allows them to be used in the industrial production of cheese and yogurt. In addition, human muscle cells will resort to this process when the oxygen supply is running low, most typically during prolonged periods of strenuous exercise, when glucose catabolism is going faster than our ability to breathe enough oxygen into the bloodstream to sustain the activity. In such a case, the cells will switch over to fermentation, allowing for a brief burst of additional energy production. But the lactic acid that builds up in the muscles during this activity causes muscle fatigue, which limits the duration that this activity can be sustained. So to summarize, all of the forms of energy production that we have learned, whether aerobic or anaerobic, begin with glycolysis. This is therefore the most evolutionarily ancient method of ATP production, and must have evolved in very early forms of prokaryotic life, given that it occurs in the cytosol and not in any membrane-bound organelle, such as mitochondria, which did not exist until eukaryotes came about. This makes sense with what we know about Earth's early atmosphere, which did not contain oxygen until the evolution of photosynthetic cyanobacteria, which prompted large-scale oxygenation of the atmosphere, making the evolution of aerobic respiration possible. In every living organism, once glycolysis is complete, pyruvate may be reduced through lactic acid fermentation or broken down and then reduced through alcohol fermentation. It can also be fed into the citric acid cycle to continue with aerobic respiration or anaerobic respiration if something other than oxygen is available to act as an electron acceptor in the absence of oxygen. In all of these processes, the NADH that forms when NAD+ is reduced during glycolysis must be oxidized to regenerate NAD+, and this is achieved either when NADH reacts with pyruvate or acetaldehyde during fermentation, or when NADH dumps electrons into the electron transport chain during respiration. The main difference is the electron transport chain drives oxidative phosphorylation, which produces most of the ATP generated during cellular respiration, whereas fermentation produces only 2 ATP, given that it is comprised only of glycolysis, so it's a huge difference in terms of energy production. And with that, we have a more thorough understanding of the various ways that cells can generate ATP. Thanks for watching. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me.
Transfer.
Professor Dave Explains at gmail.com.
Okay, so that's fermentation. Last slide na yata ito. Since hindi mag-transition. Okay. So that's fermentation. So fermentation could be done in 2 ways. So either in lactic or in alcohol, where at the end product, you will have lactic acid or either an alcohol sa fermentation process. And during the fermentation process, usually we use the yeast for food fermentation. So from the pyruvate, it depends on how which bacteria would interact. Like for example, if it is E. coli, it produces acetic acid, lactic acid, succinic acid, or ethyl alcohol. Or in Saccharomyces or yung mga yeast, it produces only ethyl alcohol and carbon dioxide. If Clostridium naman, it uses up pyruvate to produce butyric acid, butyl alcohol, acetone, isopropyl alcohol, carbon dioxide, and hydrogen. While in Propion bacterium, it produces propionic acid, acetic acid, and carbon dioxide. While Streptococcus bacillus, it produces lactic acid. If interact with Enterobacter, we have the formic acid, ethyl alcohol, lactic acid, 2,3-butanediol, and carbon dioxide. So, different microorganisms or bacteria, or if ito hindi mo to bacteria, this is a fungi. So, produces a different end products. So, it depends on what type of microorganisms that will convert the pyruvate. So let's proceed on bacterial genetics. We know that the bacterial chromosome contains double-stranded molecules of DNA arranged in circular form. And it has a length of 1,000 microns. And the DNA contains about 4,000 kilobases. Muli ang ginagamit pagdating sa PCR. So, sa PCR, 4,000 kilobases ang signaling after gel electrophoresis. Kung naa siya signal dito, once struck by UV, kung na-under siya sa 4,000 kilobases, then there is a bacterial DNA. So 1 kb is equal to 1,000 base pairs containing yung mga AT and GC nitrogenous bases. Humans have 3,000 kilobase pairs. Now, to differentiate between the DNA of the eukaryote and prokaryote, if you remember that prokaryotes don't have introns. So they have only the exons that carries the coded gene, carries the character of the bacteria. Now, sa bacterial genetics, we have here two main topics. We have the gene mutation and gene transfer. Now, sa gene mutation, we have here the types of mutation happens within the bacterial DNA. And in terms of gene transfer, this is how the DNA will be transferred sa bacteria. So could either be transformation, transduction, and conjugation. So let's have the gene mutation. Now, mutation of bacterial DNA, like in eukaryotes, could be triggered either within the cytoplasm or could be triggered in by chemical means or external factors. So mutation could happen either one base pair is exchanged for another DNA molecule or through insertion or through deletion or rearrangement of nitrogenous bases within the strand. Because like the eukaryotes also, the genome of the bacteria also move. Naga-exchange, naga-move na siya, which causes mutation. So mutation happens through asexual binary fission. So the production of new bacterial cells is not on sexual but asexual. Since they don't have gametes, the cell only divides by itself into multiple cells, so binary fission. Now, what are the mutations? So mutations, like I said before, from deletion. For example, in this mutation type of deletion, you have here the normal codons. So codons is made up of three three nucleotides. So we have three combination, AAT, AGT, GCC. Now, when deletion, tingnan natin dito. So ATT coded for leucine sa normal, AGT for serine, and ARG for GCC coded for arginine, a type of amino acid. Now, next we have the mutant. What happens? So, kita natin itong pencil. So, ito yung normal. Okay. I-blue natin. Okay, what happened? Nawala itong A. So, na-delete ang base na adenine. So, what happened? Ang mangyari, magkaroon ng rearrangement. So, nagkaroon ng ATT for leucine, GTG. So, may iba na ang coded for amino acid. That will be for valine and CCA. That is for proline. So, those are examples of mutation through deletion. Kahit deletion lang, pwede siya deletion with one base or pwede din tatlo ang mawala. And that will code a different amino acid. Another mutation type, that is insertion. So when we say insertion, like for example, we have here the normal sequence. So we have the tricodon AAT for leucine, AGT for serine, GCC for arginine. What if kung merong insertion, another nucleotide will be inserted, like AGT, AGT, ito yung AGT, ATT. Ang na-insert dito kay si C. Ito, nag-insert siya. Cytosine. So the cytosine inserted between thymine and adenine. So what happened? So there will be a change in message. Kung basahin ito ng transfer RNA, i-interpret niya ito ATT, that is for leucine, CAG, that is another amino acid, dili na serine. Then itong PGC coded for the cyst. So magkaroon ng mutation. So there will be a code for cyst formation. So once this protein will be produced, kaning protein mag-produce siya of cystic formation sa tissue. Okay. Another naman is inversion. Inversion is the DNA, like for example here, ADE. ADE. Ang inversion parang nag-invert. Supposed to be ABC is, dapat ang linya nito ganito. May nawala sa diagram. Wait lang ha. Ang original chain nito supposed to be F, F, FED. FED, then CBA. CBA. Ayan. So, for example, sa inversion, sa original chain niya, supposed to be dapat FEDCBA. Pero nagkaroon ng bending of the gene, so there will be an exchange of genome, na exchange of nucleotides or a portion or the fragment like CE na CF napunta kay aso bang first chain ani F. CF galing sa first part ng napunta siya sa end part. So nag-exchange siya between A and F. So as a result, si A supposed to be nasa last, na siya sa una part sa genome, and si F supposed to be nasa first part siya ng genome, nandun na siya sa pinakahuli. So nagkaroon ng exchange, that is inversion. Ang translocation naman is another type of transfer DNA in which nagkaroon ng from two strands, it happens between the two strands of DNA or chromosome. Chromosome siya, chromosome. Okay, na may kayasma. So originally A, B, C, D, E, F from one strand of chromosome, and the other one is G, H, I, J, K, L. And it turns out na kaning sa J o K, L, nato siya sa first strand, na-transfer siya. So ang first strand, okay, start na sa translocation. Mura siyang na-relocate. So originally, we have two strands. So strand 1 and strand 2. Okay, strand 1, let's say the A, B, C, D, E, F represent the nucleotides or the codons. Muna original. This is strand 2, we have G, H, I, J, K, L. Now, there's a translocation on the one end sa mga chromosome. What will be the end product? So, katong A, B, C, D, E, F, dili na D, E, F ang the end part, but J, K, L. Ang J, K, L, gikan siya diri sa strand ng GH. Ang I, D, E, F, gikan siya sa strand ng ABC. So, that is translocation. That is another type of transfer of DNA. So, those are some of mutations happens in the DNA. So it could either be deletion, insertion, or some may substitute like replacement of one a group of amino acid replacing another type of acid that performs the same function. Siya. So let's have the mechanism of gene transfer. Now we have three mechanisms of gene transfer happen in the bacteria. We have transformation, conjugation, and transduction. On the first, after this slide, we have discussed the first, yung transformation, followed by transduction, then conjugation. So let's have transformation. Transformation is defined as a transfer of genetic information through the activity of DNA. So this is demonstrated by the experiment P. Griffith through the mice injected with live non-capsulated pneumococci. And with heated killed capsulated pneumococci lead to the death of mice with isolation of live capsulated pneumococci. This means that some factor from dead pneumococci transferred to live non-pathogenic pneumococci. So, mura siya ang previous bacteria, non-capsulated, then the DNA was transferred to the non-pathogenic pneumococci. So, paano nangyari ang transformation? This is the process.
Transformation is the process by which DNA is introduced into bacteria. Bacteria can then replicate and express that DNA, which can be isolated for downstream applications. Artificial transformation of bacteria involves producing competent cells capable of exogenous DNA uptake. There are two primary methods of artificial transformation used in the laboratory: chemical transformation and electroporation. It is thought that chemical transformation, which requires chemically competent cells, uses divalent cations to increase the permeability of the bacterium's cell wall, thereby increasing the likelihood of DNA acquisition. Many common protocols include a heat shock step to improve DNA uptake. Electroporation involves the application of an electrical field to the competent cells in suspension, thereby introducing holes in the cell's perimeter. The newly compromised cell wall allows exogenous DNA to pass into the cytosol of the bacterium. Outgrowth of bacteria following transformation allows for repair and recovery of the cells in liquid medium without antibiotic. After outgrowth, the culture is spread on selective plates to isolate transformants containing the newly acquired DNA, which conveys antibiotic resistance. A variety of competent E. coli types are available in different formats and for different applications, such as high-efficiency cloning and protein expression. Visit clonewithneb.com for the full list of products available for this application.
So that is transformation, in which we added a new DNA fragment towards the bacteria. So there are two ways, either chemical or electro. Ano yung dalawa? Chemical at saka electroporation, in which i-change mo yung charges ng membrane in order the DNA could enter to the cell of the bacteria. So meron kang i-introduce na DNA. So this kind of method is essential or beneficial in producing medicines or even used in gene therapy. How about transduction? Now, transduction generalized involved in any segment of DNA, restricted when specific bacteriophage. So this is a different way to insert DNA to the cell. We use bacteriophage. So we need a virus to insert its genetic material towards the bacteria. So transduction affects plasmids and episomes. Plasmid transfer induces penicillin resistance in Staphylococcus, helps genetic mapping. Also, eukaryotic cells helps. This is also helps in genetic engineering. So, nandito na yung mga GMO products sample. So, let's have transduction, how it was being done.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the gal gene, near the specific site of insertion remains attached to the phage DNA and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
So, the difference between transformation and transduction, we use a virus to inject the DNA, genetic material towards the bacteria. Now, let's proceed with conjugation. Transfer. Okay, conjugation is a natural way where the bacteria multiply itself by before binary fission. So during conjugation, there's an exchange of genetic material. It requires two cells of bacteria. Then a pilus will be developed between them as a bridge in which they're going to exchange their plasmid. So what was used in this process are the plasmid. Plasmid is another genetic material carried by the bacteria. And this plasmid also responsible for the resistance to different drugs. So kaning plasmid anong usually nagamitate for the bacteria to become resistant to other chemical antibiotics. So let's have conjugation.
Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of two different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.
So that's all for transduction, transformation, and conjugation. Any questions?
Wala po, ma'am.
Wala?
Wala po, ma'am.
So medyo klaro-klaro ang topic?